that entire story that you gave us, can you give us an idea of what we can do with that? What is necessary to get on board with this? Is it pride? Is it protocol? Is it structure? I mean, what can we do with it? Because I think that's what ultimately everyone wants, just like we had gotten polio rid of and measles and some of these other things. What is one thing that we could get rid of cancer by just not ever letting it be? I mean, that's what we achieved with other diseases.
Thank you, Sanjay. Well, you said so many interesting things. First of all, I loved that documentary of AlphaGo. I watched it many times, and Demis Hassibis obviously got the Nobel Prize as a result of just exactly starting that. That's where he got his go. And I think that it's a very important example that you just gave, that the fact that we have ourselves created a machine that can teach itself to think 70 steps ahead. Humans can only think 2-3 steps ahead. I mean, if our IQ is the highest recorded is 176, the IQ of a regular AI thing is 2000, and that of super ones is going to go into millions. So how can we compete? But we have created these machines that can do this stuff for us. So, for example, I mean, I'll keep coming back to this, to the device to say that it doesn't just detect cells. It can detect biomarkers such as what the cells are shedding, pieces of DNA, pieces of RNA, pieces of protein. So peptides can be detected by using single molecular electron field transistors, can detect all these biomarkers. You see, the difference is that Grail is using AI right now to enhance their machine learning capabilities, and I just told you that they've become so much better. But their problem is they're still dependent on periodic testing, once a year or twice a year at most. But what I'm saying is that while you're waiting for that 1 year or 6 months, you can develop a cancer that then requires slash, poison, or burn. So what you need for continuous monitoring, this device that I just showed you can sample the entire 5 liters of blood for all these biomarkers and any abnormal cell within 18 days. The entire body's everything will be. So imagine this thing is sitting in your blood vessel, constantly monitoring. Every 18 days, every bit of your blood has been monitored or has been screened for any abnormality. And I'm saying that these are the kinds of implantable devices that are going to have to replace all kinds of disease detection. Because what we are aiming for is to detect that wellness to illness transition, even before the transition point actually happens, just the whisper of it. And at that point, fixing things will be so much easier. You could probably have a health drink which is supplemented with the exact right kind of antidotes that you need, or a simple lifestyle change could make a difference at that time. So I feel that having the kind of technology that's available, and I've been a miser in one sense, although I've published hundreds of papers using so many of my samples, but I only use samples where I have duplicates available. Because I've been saving samples for the one grand study we'll be able to do. Now technology has arrived. So now I can take serial samples. Some people who lived for 10 years, some who died within 6 months. I have serial samples on both. But the most important is the pre-diagnosis sample on each. Could I have predicted that this one is going to survive 10 years and this one is going to die within 6 months? And if yes, what do I need to do with it? Well, right now what we can do is perform multi-omics, which is, genomics, transcriptomics, metabolomics, proteomics, multi-omics on these samples. And we figure out that this is the combination of signatures that tells us that things are going to move aggressively in this person. If we have that kind of a signature, even before this implantable device gets approved by the FDA, we can start using those kinds of signatures not once a year or twice a year, but with point of care technology. You know what that is? That is that, again, biomedical engineers, Professor Sam Sia at Columbia has built literally a machine that's the size of a toaster, which can perform PCR, RT-PCR, all kinds of things on a drop of blood. So sitting at home with this little cassette that you put into this toaster-like machine, you can test yourself every week, every day, every month, whatever you want for this signature, while we are waiting for the implantable devices to be approved. I'm saying the problem is, Sanjay, no one wants to think like that. No one wants to think of, I am desperately trying to raise resources and funds to help me and my colleagues, the biomedical engineers, to get this thing to the bedside. We are saying that given my tissue repository, well, basically, they have developed this device. But what should the device detect? I have that. Cells and the biomarkers. That's why our complementary strengths are so important. Do you know we are claiming that within 6 to 8 months, we can have all the biomarkers identified because we have all the sample. I just have to mail it or just take it down to the Columbia's core facility for doing the multiomics, and they'll give me within 3 months. And then I spend 3 months analyzing the data to come up with the signature. And they just need to finalize something points in their device so they can start putting it in large mammals. Given the right resources, we can do this in 6 to 8 months, but no one is willing to give the resources. I have been groveling in front of billionaires, in front of big pharma company, in front of academic leaders, everybody. No one wants to listen to me because all the money is in the other aspect, the last cell and the treatment developments. So you tell me, Sanjay, you interview a lot of people. Who do I go to? Who needs to listen to me?
I've had to experience the very quick disillusionment and harsh reality of healthcare in this country is just different than it is elsewhere. I mean, like you don't make progress and productivity, one, by not knowing it, two, by acknowledging and harping on it, but three, the way you do it is to accept it and thread the needle, which I know you know well, and unfortunately it sounds like have laboriously appreciated. I think one place that we could actually really kind of find a proof of concept, and before I say this, I need to orient for anyone listening. When y'all hear biomarkers, and when you're hearing about this signaling on the before space, I actually just watched a Netflix show, documentary called Inside the Mind of a Dog or something like that. And apparently dogs, when they smell in 3D. So like where we can tell when we hear where something is, an idea, even with your eyes closed, on is it in front of you, behind, but to the left, to the right, in 360 degrees. They can apparently do that with smell. And that made me immediately think back to when I first was told that dogs can predict seizures and they can actually predict cancers potentially or death. And I was like, that's nonsense. There's no way. They're not like, spiritual or on some different plane of time. But really what the science is showing is they're able to smell the byproducts of this very complicated but highly reproducible and orchestrated system that is the human physiology and biology. So like on the skin, the discharge elements, the electric stuff, the catecholamine releases and what that does, all of this is so beautifully harmonious and dynamic that if it can do it by smell, you're talking about something in that same capacity when it comes to environmental orientation, landscape signaling, the same way a fire is there with the smoke. You don't have to see the fire, but the smoke is there, and all of these things, the way we can predict wildfires and everything. You're saying, Sanjay, and if anyone doesn't know this, all of this circulates. The blood is literally the room with which sound travels. It's the same thing for everything in your body. Your entire blood system goes through every single cell in your body. I mean, that's a fact. This is the highway. And so you're saying, let's analyze the highway for something that flows into every cell that we have and see when the callus formation is happening on the foot compared to like the baby soft foot, or when, when all of these things happen when it comes to these changes before it's too late, like the leg discrepancy. All of that to say, what I'm thinking is, instead of nixing it forever, one area could be potentially where you have patients that have been cured of cancers. So you already have a head start on what changes dynamically happened, if you're lucky, if you had enough samples that it just didn't walk in and one day are diagnosed, because then it's useless, right? Except for what you have today. But if you have a serial idea and AI can say what mutations likely occurred over what period of time to eventually, quote unquote, get here, get unregulated, which is what cancer means. And then you apply this in these stage 2s or stage 3s, not necessarily for recurrence even, but even the ones where we know second primaries happen, which that means that there are cells that are along the process of becoming a cancer, whether invasive or not invasive, but there's steps along the way, meaning tubulovillous adenoma versus, something that's less evolved. Now you have a narrowed scope of the 2 or 3 steps remaining before it becomes cancer. So it's not necessarily for the recurrence of the same previous one, even though I could conceive an area where that makes sense. But the second primaries, the colon, the lung where smokers happen, like lung cancer patients get a different one because they've already had a few of those steps, in the area and in the colon. The rest of the conversation you and I are going to have offline. But certainly, I think these are ways that one can thread the needle, because you can have a therapeutic intervention and quickly, because you're able to say, aha, the same thing that made it HER2 positive, or the same thing that made it, whatever the case may be. There's still that economic sad reality. But 2, it will give us an idea of then hopefully how to avoid those second primaries, which will be preventative. I don't know. That's me thinking out loud and in real time. But I just hope that by listening to you and your insights and your collective, aggregate of observation and now fervent need for action, that anyone can appreciate this is a dynamic evolutionary process. We don't get enough pictures of the evolution. We have a gazillion sad examples, the aggregate that is the tragedy of cancer and suffering. We have all of that out there, and we're not doing anything to serially monitor it, because we know that we have clues that happen before we get there. We know that. And yet we're like, meh, we'll wait till we get there. And that's maddening. So, whereas a lot of these, podcasts will have like what needs to happen in a technical aspect or regulatory or whatever. This one's scary, but so large in its return is, how do we get people on board with what we know to be true? And that is the number of steps you have to get to third base before you get to home base in baseball. There is no rule where you're going to get to first and then home. That's just not possible. And the same thing's for cancer, and yet here we are.